Hi everyone, my name is Anna, and today I'm going to talk to you about adult brain plasticity and how we can make use of it in autism spectrum disorder. So, autism spectrum disorder is a group of neurodevelopmental disorder that affects approximately 1% of population. It is characterized by three main behavioral symptoms, such as social communication deficit, impaired social interactions, and engaging in repetitive or restricted behaviors or interests. Adults with ASD often experience poor educational and employment outcomes, as well as lower IQ levels as compared to age-matched groups. And that was basically the rationale for our study. So rather than curing autism, we wanted to find ways to improve functioning in adults with autism. So the causes of autism can be categorized into three different groups: genetic, environmental, and the majority of cases are idiopathic, meaning without a known cause. And researchers to study autism often use different mouse models, and as you can see, each group have a corresponding mouse model. So in our study, we used CADNAP2 mouse model, which is a genetic mouse model, and BTBR mouse model, idiopathic model. I don't know if any of you are familiar with animal models. This is quite a controversial topic, so I just wanted to briefly mention that all of the studies are regulated by law, and researchers have this golden rule to follow, which is called 3 Rs. So we are trying to reduce the number of animals used in our studies. We are trying to find replacements for animals when possible, and we're trying to refine the conditions in which animals are being tested. So obviously, it's not possible to find one brain region that would be responsible for all the behavioral impairments seen in autism. However, there's one of a particular attention for our study, and that is hippocampus. And here on the slide, you can see how hippocampus looks like under the microscope. It's just a brain slice of rodent's hippocampus. And as you can see, it consists of 4 parts: the dentate gyrus, here, the CA1, CA2 here in the middle, and the CA3. So in our study, we focus mainly on the CA2 and the CA3. CA2 is a fairly understudied region, and research studies show that it's involved in social behaviors, mainly in the social memory. The CA3 is responsible for cognition. So it was also of a great interest for our study because of behavioral symptoms connected with autism, which are social behaviors and cognition. And finally, the dentate gyrus, which is a place of plasticity. So what exactly is plasticity? It's brain's ability to adapt to changes. And in our study, we focus mostly on one of the aspects, which is adult neurogenesis. Adult neurogenesis, so for decades, people thought that our brains, when we grow older, is not able to grow new neurons. However, in the 60s, people challenged this finding, and right now we know for a fact that our brains can grow new neurons, even if we are old. And here on the slide, in pink, you can see the adult-born neurons from a rodent brain, so we can actually see that the population of adult-born neurons is quite big. And since adult neurogenesis is one aspect of brain plasticity, it can be regulated by experience. So we can distinguish factors that can increase neurogenesis, such as physical exercise, environmental enrichment, and sexual experience. But on the other hand, we also have factors that can reduce adult neurogenesis, such as stress or obesity. So why do we really need these adult-born neurons? They've been implicated in a number of behaviors connected with memory and cognition. That's why we could assume that adult neurogenesis could be implicated in the pathophysiology of autism. So what I did for my study is I used the BTBR mouse model to check whether the adult neurogenesis is reduced in this mouse model, because other studies show that other mouse models of autism have reductions in adult neurogenesis. So I labeled new neurons that you can see here in red again, and I quantified the number. And what I found is that adult neurogenesis is indeed impaired in the BTBR mouse model. And adult-born neurons also are very important because they make functional connections with other brain regions. So they send projections to both the CA3 and the CA2, which are implicated in pathophysiology of autism. So I wanted to see whether the reduced number of new neurons means that there's less connections to those brain regions. And here in red, on this photo, you can see the axons coming from those neurons. And this is just a photo of control group. And after performing the analysis, I saw that there is much less connections of adult neurons coming to both the CA2 and the CA3. The next form of plasticity that we've been investigating is called perineural nets. Perineural nets are those tiny structures that form lattice-like nets around different neurons in your brain. And basically, they develop as we grow older, and by the time when we reach critical period of our development, they're fully established. And they basically limit plasticity in our brain. But interestingly, studies show that if you get rid of the nets in your brain, you can restore the juvenile-like plasticity. So the adult brain can actually have plasticity of a young brain again. And why are they really interesting in studying autism? Well, studies show that the same genes that regulate the expression of perineural nets are also involved in the pathophysiology of autism. However, no one ever looked at their expression in autism. So we decided to look at the perineural nets in the brains of mouse models of autism. And what you can see here is the CA2 region that you can see probably here as well. So the same region responsible for social behaviors. And you can see how the volume of perineural nets in these mouse models of autism is increased. So basically, it means that the region with this increase is becoming less plastic, more rigid, and it can form less connections with other brain regions. Next, just using the marker of the CA2 that you can see here in bright red, we wanted to see whether the area of the CA2 covered in perineural nets is going to be different. And actually, mouse model of idiopathic autism shows that the percentage of CA2 covered in perineural nets is much more bigger. So there's a bigger area without plasticity in this model. And now just take a minute, we should just take a minute to combine those two aspects. So this is the hippocampus that I showed you before, and this is how you can see it with all the cells in it. And here in dark blue, you can see the population of newborn adult neurons. And as I said, and this is the CA2 with perineural nets. And as I said, those neurons can make functional connections with the CA2, strengthening the function within the CA2 region. And this is what is happening in a normal control brain. The connections, there's a lot of connections and they're not disrupted. However, when we take ASD brain, we can see that the population of adult neurons is much smaller. And the intensity of perineural nets in the CA2 is much bigger. So there's more of perineural nets and they're more mature. So that could possibly disrupt the connections. So maybe the adult neurons are not really able to make connections with the CA2, and that's why we see social behavior impairments in those animal models. So the next thing that we wanted to do in the lab, and that is happening right now, are manipulations. Are manipulations. So the first step of the experiment is to perform behavioral analysis. So basically, mice are undergoing two types of behavioral tests. The one is to check the social behavior, and the other one is to check cognition. So in the first one, you have the mouse put in the middle of the apparatus, and you have two rooms, one with its cage mate and the other one with a novel mouse. And we would suspect that the normal mouse is going to spend more time with a novel, that the tested mouse is going to spend more time with a novel mouse, because that's something new and exciting, and they're not really interested in spending time with their cage buddies because they know them quite well. However, if you take the mouse model of autism, these mice don't really remember their cage mates. So they would tend to spend the same amount of time with both of them, meaning that they have impairments in social memory. And the same with cognition. You just let the mouse explore two objects in acquisition phase, and then you perform the test in which you move one of the object. And you would expect the mouse to discover more the object that has been moved. However, mouse models of autism don't really do it because they can't recognize the change. And we performed those tests, and we actually discovered that mouse models of autism have impairments in both of them, meaning that they have impaired social memory and impaired cognition. And the next step would be to perform 2 forms of manipulation. The first one is to reduce the number of perineural nets, which can be done with just injection of an enzyme directly to the brain. And this injection can be specific, and you can just pick the area that you wanted to inject. So you can get rid of perineural nets in the CA2 for up to a couple of weeks. And the other manipulation is to increase the number of new neurons simply by physical exercise. So you just put running wheels in the cage for 6 weeks, and mice can actually grow more new neurons if you let them run. And after those experiments, they're going to undergo the same test once again and see if the behavioral impairments improve. So combining those 2 methods could actually show that that the behavioral impairments could be potentially reduced in those mouse models of autism, and maybe using perineural nets and adult neurogenesis could be a way to treat symptoms in autism. So I just wanted to acknowledge my team from Princeton that we're working on the same project. And thank you for your attention.